Neurons are going to be considered really the structural unit of the nervous system. So they're really the main group of cells in our nervous system, functioning to provide that control or helping us maintain overall homeostasis. So neurons are also known as our nerve cells. They have a couple of key characteristics. First, neurons are excitable. If a cell is excitable, that means that it can rapidly change the voltage or the ion concentration or the charge difference across that plasma membrane. So neurons are excitable. That means they can rapidly change the voltage or the difference in charge across that plasma membrane. That difference in charge across the plasma membrane is known as the resting membrane potential. So neurons can rapidly change that resting membrane potential as a means of communicating with one another. Neurons are also amitotic. So amitotic means that they do not have the ability to divide. It's important to realize that there are exceptions to this rule. So although we're normally going to say that neurons are not going to be able to be replaced if destroyed, there are some exceptions to that rule, so just be aware of that. Neurons also have extreme longevity. So assuming that they have a steady nutrient supply, they can actually function pretty normally for a lifetime. So they have extreme longevity, or I guess you could say a long lifespan. Neurons have a really important function for control, right? So they are the structural unit of our nervous system, and so they're very active cells. That means that they have a very high metabolic rate. So they require a continuous supply of oxygen and glucose in order to generate ATP to fuel their cellular processes. So neurons have a high metabolic rate, which just means they're very active cells. So they need a continuous supply of oxygen and glucose in order to generate ATP to fuel their cellular activities. We're going to take a look at the structural classifications of neurons, and you'll see really clearly that neurons also have a lot of variability in their shape or their structure. So if I flip forward here, we're going to cover this a little bit later, but you'll notice that there's clearly a lot of variability in the structures of our neurons. We have what are called multipolar neurons, we have bipolar neurons, and we have unipolar neurons. So big characteristics of neurons: they're excitable, they're amitotic, they have extreme longevity, they have a high metabolic rate, and they have variable shapes. So we're going to first just talk about the neuron structure in general. Again, keep in mind that the neuron's shape can really vary. However, all of our neurons are going to have the same three basic components. They're going to have a cell body, and then two different types of cellular processes known as dendrites and an axon. So a neuron is composed of a cell body and then two different types of cell processes or extensions known as dendrites or an axon. So this is our neuron cell body. The neuron cell body is going to have all of our organelles and our nucleus. So it's going to contain the nucleus and then the cytoplasm. So again, the cytoplasm is going to be everything that exists outside of that nucleus. So that's going to include all of our organelles outside of the nucleus, as well as the intracellular fluid or the cytosol. Because neurons are extremely active cells, or they have a high metabolic rate, you'll notice that the neuron cell bodies are abundant in mitochondria. There's a bunch of mitochondria present in those neuron cell bodies. That's because that's where we're going to generate the majority of the ATP needed by these cells in order to fuel their cellular processes. Protruding out from our cell bodies, we have these processes or protrusions. These are known as dendrites. So dendrites are also going to have cytoplasm present within them. So very similar cellular contents as the cell body. But the dendrites are these cellular protrusions that really function as our receptive regions. So dendrites are going to be our cellular processes that are actually receiving information from other neurons. So you could also call these dendrites input regions because they might be receiving input from other neurons or receiving signals from other neurons. You'll notice that we have many dendrites or many of these kind of protrusions out from the cell body. That's really increasing our surface area or maximizing our surface area for receiving those signals from other neurons. So dendrites are then going to convey any information that they're receiving to the cell body. And then neurons are then going to convey information away from the cell body via an axon. So neurons receive information at the dendrites. They're going to send that information to the cell body. And then information is going to be sent out from a neuron via our next cellular process known as an axon. So an axon is almost this tail-like structure coming off of the cell body. The region where that axon comes off of the cell body is known as the axon hillock. And then we have this long axon tail. Each neuron only has one axon. However, that axon is capable of branching in order to innervate or synapse with a variety of other cells. So each neuron only has one single axon, but that axon could branch a number of times. So axons are going to be sending nerve impulses away from the cell body. They're going to be sending those nerve impulses towards axon terminals. Axons could be surrounded by what's called a myelin sheath. A myelin sheath is also known as that insulating cover. The myelin sheath acts as electrical insulation. This is allowing for nerve impulses to travel faster down that axon. So it increases the speed at which neurons can send nerve impulses or communicate nerve impulses. The myelin sheath is going to be composed of layers and layers of phospholipid membrane. This phospholipid membrane is coming from either Schwann cells in the peripheral nervous system or oligodendrocytes in the central nervous system. So Schwann cells are going to wrap entirely around that axon of a neuron. So the entire Schwann cell is going to wrap around that axon. Whereas oligodendrocytes are going to have those cellular processes or appendages that are going to reach out and wrap around an axon of a neuron. So it's not the entire oligodendrocyte that's wrapping around that axon. It's actually just one of its appendages or cellular processes. So we can see the myelination in the peripheral nervous system in this diagram here. So we can see that you've got a Schwann cell, and the entire Schwann cell is actually wrapping around that axon. As that Schwann cell is wrapping around the axon over and over again, you'll notice that all of the cytoplasm in the nucleus is being pushed out to the outer edge of that myelin sheath. So you can see again that as this cell is wrapping around a number of times, eventually all of the cytoplasm in the nucleus is being pushed to the outside edge. So now we just have layers and layers of phospholipid membrane, or layers and layers of the Schwann cell plasma membrane, forming this phospholipid insulation around that axon. So the myelin sheath is then going to be composed of these layers of phospholipids. And then on that outside edge of the myelin sheath, we're going to have all of the cytoplasm in the nucleus that was pushed out to the outer region, so that then we could have really tight layers of phospholipids really close to that axon. So that's what gives this the insulation. So we've got a bunch of tightly packed phospholipid membrane layers in the myelin sheath. Then all of the cellular contents were pushed to the outside edge. And that's going to make up the outer collar of the perinuclear cytoplasm. So the outer collar of the perinuclear cytoplasm is going to consist of the Schwann cell cytoplasm in the nucleus. Again, as that Schwann cell wraps around a number of times to get the really tight layers of phospholipid membrane, the cytoplasm and the nucleus get pushed to the outside edge, forming that outer collar of perinuclear cytoplasm. We do not see this outer collar in the central nervous system because the myelin sheath is going to be formed by oligodendrocytes as opposed to Schwann cells. So in oligodendrocytes, we have these cellular processes that are wrapping around the axon, but we still have a cell body over here that's far away from that axon. So the cellular contents don't have to get pushed to the outside edge of this cellular process. Instead, all of the cytoplasm in the nucleus exists in the cell body out here, and it's just that small appendage that's really wrapping around that axon. The myelin sheath is going to have these gaps in between the Schwann cells, or in between the processes of our oligodendrocytes. Those gaps are known as the nodes of Ranvier. So the nodes of Ranvier are also known as the myelin sheath gaps. And action potentials, or nerve impulses that are being sent through this axon, are essentially going to jump from one segment to the next, right? So they're going to jump from one myelinated section to the next in order to keep that really fast transmission. Non-myelinated fibers are also found in the central nervous system and the peripheral nervous system. So in other words, there are neurons that do have axons that lack Schwann cells and lack oligodendrocytes, so they do not have as fast of transmission of their nerve impulses. So in our CNS and our PNS, we have both myelinated fibers, which have that myelin sheath. We also have non-myelinated fibers, which lack that myelin sheath. That myelin sheath is not only going to play a role in providing an insulating covering in order to speed up the rate of transmission of nerve impulses, but it's also going to play a role in regenerating or repairing damaged axons. So the myelin sheath is important for speeding up the rate of transmission of an action potential or a nerve impulse, but it also helps with regeneration of axons in the peripheral nervous system. So if an axon gets damaged, it is not going to be fixed, or that neuron's not going to be replaced in the central nervous system. However, in the peripheral nervous system, damage to the axon actually can be repaired. So if an axon was damaged in the peripheral nervous system, so let's say that, for example, maybe you broke a bone and one of your peripheral nervous system axons was severed in that process, Schwann cells, which make up the myelin sheath in the peripheral nervous system, can actually regenerate that axon by forming what's called a regeneration tube. Oligodendrocytes cannot form this regeneration tube. So if an axon of a neuron is damaged in the central nervous system, it's then not going to be a functional neuron, so it's not going to be replaced or repaired. So we can see that brief process here for how our Schwann cells are able to regenerate an axon. So really briefly, if there was fragmentation of that axon, or if this axon had severed here, the cut axon ends are then going to seal themselves off. So essentially we're going to have kind of two pieces of an axon, the more distal region containing the axon and the axon terminal, and then the other side, which is going to be the axon that's connected to the cell body. So the cut axon ends essentially seal themselves off. So now they're completely separated from one another. Schwann cells and then resident macrophages are then going to come in and they're going to clean out that site of injury. So they're going to clean out any neuronal debris. Schwann cells are then essentially going to form this regeneration tube, which is basically just Schwann cells surrounding that site of damage. And they're going to start secreting growth factors and what are called cell adhesion molecules that are then going to encourage that axon to grow and actually recombine with the other cut or sealed end of the axon. So the Schwann cells are going to wrap around that site of damage, forming a regeneration tube where they're secreting growth factors, they're secreting cell adhesion molecules that are really trying to promote the axon, the damaged ends of the axon, to essentially connect to one another or re-adhere to one another and then regrow. And then eventually that axon that's regenerating is then going to produce a new myelin sheath. So this diagram, I think, also does a nice job of showing the formation of that regeneration tube. So once again, if you have injury to an axon, so for example, that axon is severed, the cut ends essentially seal themselves off. Schwann cells, macrophages are going to come in, they're going to clear out any neuronal debris. And then those Schwann cells are going to form this kind of regeneration tube. So they're going to start secreting growth factors as well as cell adhesion molecules that are then going to promote the adherence of these two regions of the axon, or of the kind of two severed ends of that axon. So now we're going to have this regenerating axon coming up, and we have the new Schwann cells forming along the growing axon, and we have reattachment of those two ends of the axon. Then we have an entirely new myelin sheath that's then going to form over that regenerated axon. Once again, axons are not able to be regenerated in that central nervous system. That's predominantly because we have a different cell in the central nervous system forming the myelin sheath. So oligodendrocytes cannot form that regeneration tube, and so we cannot actually repair a damaged axon in the brain or the spinal cord. So just to provide a little bit of clinical relevance for those myelin sheaths. So in multiple sclerosis, there's actually demyelination due to inflammation. So for whatever reason, there's going to be this inflammatory response that is damaging those myelin sheaths. And so in multiple sclerosis, we actually see a change in the amount of white matter and gray matter in the central nervous system. And so when you're visualizing the central nervous system using an MRI or histological sections, you'll see that the central nervous system has matter that looks more white. That white matter is going to be the myelinated fibers. Myelinated fibers are composed of all those phospholipids, which don't stain very well for histological sections or MRI, so they appear white. Whereas the gray matter would then be all of our cell bodies and dendrites. They do stain well, so they are visualized as gray matter in histological sections or MRI. And so in multiple sclerosis, you notice that there's essentially a decrease in white matter because we have demyelination occurring. So that myelin sheath is being degenerated by inflammation.